Good morning! Thank you guys for getting up to listen to the Fast Science Summit. And we've got a really phenomenal lineup for you guys today, so I think it's going to be an amazing day with a lot of information, and we'll just see how it goes. So what I'm going to do first is give a small introduction, and I'm calling it Genetics and Therapeutics 101, and this is just to refresh everybody on the genetics and how we're looking for therapeutics in a general sense. And then you're going to hear from the people who are actually doing this, who are doing the work and doing the research. For those of you that are out on the live feed, if you want to send questions, you can do it either through our Facebook page, look for Foundation for Angelman Syndrome Therapeutics, or you could tweet it to us either at @CureAngelman or at @fast_science, and we will try to get your questions to the people in the room. So hi to all of our live feed watchers. All right, okay. So a little bit of history of Angelman syndrome. So Dr. Harry Angelman, who's here, actually 50 years ago this month published a paper on puppet children. He had 3 cases, 3 children who came into his clinic, and they all had very similar characteristics, and they were very unique characteristics. And if you think about Angelman syndrome, it's incredible that 3 people in the Liverpool area all came to his clinic, and he noticed that they were all so similar. And as the legend goes, he named them puppet children because he had seen this picture of a boy with a drawing of a puppet, and the boy was very happy, and he thought, I'll use this as my term. Now, we don't use this term anymore, and we really ask people not to use this term. And what we do now is call it Angelman syndrome after Dr. Harry Angelman, who first described it. I think some of you also know that this painting was recently stolen just last week, I think. Hunt for it right now because we'd like that part of our legacy back. So it was a while later, 1987, when it was determined that children who had Angelman syndrome had small deletions. And we think of them as big deletions because they're big things in our lives, but they're actually really tiny. But that led the way to figuring out what it is in that region of chromosome 15 that is so important for Angelman syndrome. And in 1997, it was determined that the gene UBE3A was the causative gene in Angelman syndrome. So to think about how Angelman syndrome ends up manifesting, you have to think about genetics a little bit. And so I want to remind you that we all have 23 pairs of chromosomes in our cells. And today we're really only focusing on this pair, and this is chromosome 15. And we get one set of that pair from our mom and one set of that pair from our dad. And so in the one cell stage, you've got one set from your mom and one set from your dad, and that one cell is then going to divide many billions of times to make the person that's all sitting here in this room. And I'm a developmental biologist at Princeton University, and that's what I actually study, is how do you go from one cell to all of us in this room who have your arms and legs in the right places. And if you think about it, it's pretty miraculous. But in this case, we're talking about one set from mom and one set from dad. And the thing that's really cool about this is your cells know which set of your genes came from your mother and which set of your genes came from your father. And the way they know that is through something called methylation. And so we talk about methylation a lot when we're talking about our diagnoses. And so what I'm showing you here is a neuron. And this diagram is showing you what the genetics looks like on chromosome 15 right next to UBE3A. So in these diagrams that we always have in our science papers, this box represents UBE3A. So this is where that gene is on the chromosome. And on the maternal chromosome, the box is red because this box is actually making UBE3A RNA, which is going to make UBE3A protein. On the father's chromosome, this box is black because it's not expressed, it's silent. And the way your cells know the difference between mom and dad is this methylation. So this is a modification of DNA that the cells recognize. And the methylation modifications from mom are different than the methylation modifications from dad. And so when we've asked you, when we're talking about diagnoses, have you had the methylation test? What we're asking you is, has someone checked the DNA to see if both mom and dad's methylation patterns for this region are present? If they aren't, that tells us something about the type of Angelman syndrome that the individual has. Now, in an Angelman syndrome neuron, the maternal copy is gone. This is the deletion case. So that maternal copy is gone, so there's no expression of this gene. And then on the father's side, because of the methylation pattern, there's a different transcript that's expressed. And we talk about this transcript. It's called the antisense transcript, or the ATS. And we don't understand the mechanism completely, but because the ATS is expressed, it prevents UBE3A from being expressed. Now, I do want parents to realize that in all the other cells in your body, both copies of UBE3A express. So even if you have Angelman syndrome and you're lacking UBE3A in your neurons, all of your non-neuronal cells still have UBE3A. So this is a really interesting facet of Angelman syndrome, is that it's neuronal specific. So we know the gene that's involved, and we know the cells that we actually need to go and target, and those are the neurons. All right, so with that background, these are the known mechanisms for Angelman syndrome. So in this diagram, I'm showing you the female chromosome is in red and the male chromosome is in blue, very clever. And so on the red chromosome is the UBE3A that's expressed, so it's in bold type. And just remember, on the paternal chromosome, there's UBE3A, but it's not expressed, so it's gray because it's silent. The vast majority of us have children with deletions, and what happens in the deletion then is the UBE3A gene is just missing. It's not there in the mom's chromosome. The cells don't recognize it as being something to express in dad's chromosome, and you miss UBE3A. With a UBE3A mutation, you'll have a normal methylation pattern because it will see that both mom and dad's chromosomes are there. But UBE3A itself has had change in it that has prevented it from being functional. And if you want to know more about point mutations and how they work and how they disrupt function of UBE3A, I'm happy to talk to anyone at any of the breaks. There's also uniparental disomy, or UPD. And in this case, this is another thing that's almost like a miracle. So what happens is when you make that first cell, something happens and mom's chromosome gets kicked out. And it can't survive with only one chromosome 15. And life is a powerful selection tool, so what it does is it copies the chromosome it has from dad, and now you have two chromosome 15s. So this is what we call rescue, and the cell knows how to do it, and it does it very well. The only problem is that now you have two methylation patterns from dad, and so the gene is not expressed from either chromosome. So it does cause Angelman syndrome, but the miracle here is that you have a living person. And finally, there's imprinting center defects. And so in this case, methylation doesn't look normal, but it's not UPD. And what it is instead is it's other parts of the chromosome that are important for putting those methylations in the correct places. And that process goes awry, and you end up with a chromosome that is not recognized as being from mom, and so UBE3A is not expressed. So these are all the major ways that we have Angelman syndrome. And the thing that's important to keep in mind is that in all of these cases, there's a perfectly good copy of UBE3A sitting there. It's just not being utilized. So what is UBE3A? So UBE3A, you can think of this as part of a protein complex, which is essentially a machine. It's a little machine in your cells that has a function, and UBE3A is part of that machine. And the function of this machine, which involves UBE3A and some other proteins, E2 and E1, is to take a modification called ubiquitin and put it onto other proteins. So UBE3A in its little machine complex goes around and finds the proteins it's supposed to add this ubiquitin to, and it does that. And then what happens most of the time is proteins that have been tagged by UBE3A in this machine get taken to another machine called the proteasome. And this you can think of as a recycling center. It's another protein machine in the cell, and what it does is it takes those proteins that have been labeled and disintegrates them. It basically recycles them, breaks them up into little pieces, and those pieces get recycled by the cells for something else. And so if UBE3A is not present, proteins that should be labeled aren't labeled, and the idea is there's probably a buildup of proteins in neurons that shouldn't be there, and that somehow is interfering with optimal neuron functioning. That's how we mainly think about UBE3A function. I just want parents to understand that ubiquitin is a modification that typically targets other proteins to go be recycled by the proteasome. But there are also cases where you can add ubiquitin to other proteins in the cell and cause them to have different activities. So UBE3A is involved with recycling proteins, but it may also be involved with modifying other proteins to do their jobs effectively. But the take-home message is without UBE3A, whatever this ubiquitin labeling is doing is not happening, and it's causing neurons not to function correctly. So before I tell you about in general, how we're approaching looking for therapeutics, I just want to stop and say, this is a reasonable question. Is it reasonable to think we can even make a targeted therapeutic for Angelman syndrome? And this is something we debate a lot on Facebook. And personally, I think there are a lot of reasons to think that this is possible. And I'm just going to share 3 of them with you that are, I think, particularly compelling. The first is this paper in 2007 that came from Ipa Elgersma and Ed Weber. And what they found was that they could rescue the phenotypes of the Angelman syndrome mouse by making modifications in other proteins. And the reason that this is so compelling is the protein that they modified is a protein called CAMK2, and it's important in neurons, it's important for learning and memory. But the thing that I want you to know is that this protein is predominantly expressed in neurons after the mouse is born. So what that says to us is that the brain's developed fine, the neurons developed fine, there's nothing necessarily wrong with how the neurons were made. What really seems to be wrong in Angelman syndrome is how they function. Now, as a developmental biologist, I can tell you that if your heart doesn't form, I'm not going to be able to rescue that very easily in you if you're born without a heart. I mean, we could do a heart transplant, but it's not an easy thing to do. So if Angelman syndrome caused strong defects in the way that the brain was formed, there wouldn't be much we could do about that. But it looks like that's not the case. It looks like the brain is fine, and what we really need to do is think about trying to fix the functioning of those neurons. And this was the first paper that said it was possible, and it was also one of the first papers that came out that said you could do this for any neurological disorder, and has really set off a wave of change in the neurological community, or the neuroscience community, who used to think that there were windows where things could happen and windows where things can't, and that has all been upturned and things are really changing. The second, and this is another paper from Ed Weber's lab, if you restore UBE3A into the adult brain, an adult mouse brain, so we take a virus, go into an adult mouse and put some UBE3A coding sequence back in the brain, you can rescue some of the phenotypes. An important thing to keep in mind here is that this is an adult mouse. So this is a mouse that's had Angelman syndrome its whole life. We go back in and add some UBE3A, and now these mice have some recovery of their functions. That tells us that there's going to be a lot that's possible. And there was a recent study that suggests that there are some phenotypes that might require more early intervention than later intervention. And that absolutely may be true, but the really important thing about this paper that I want you to take away is that neuroplasticity, the functioning of those neurons, could be rescued at any age that they tried. So we think the mouse model is telling us that we have a lot of potential for therapeutics. We can also say that if you're a mouse with Angelman syndrome, we have some very good news for you because we can take care of you. But the bottom line is there is a reason to believe this is possible, and we're not going to know until we know. So we have to keep moving forward. We have to keep looking for therapeutics, and once we have them, we'll know what we can rescue and what we can't, and what we can make better and what we can't. But there's plenty of hope. All right, so general ways to find therapeutics. This is the easiest, or I would say the most upstream approach to this. And that would be trying to just reactivate that silent copy that's sitting there in all your neurons and ready to go. And there are a lot of people who are working on these types of strategies. There's reactivation of the paternal gene, and that would involve chemicals, antisense oligos, different engineered proteins, and you'll hear about some of that today. Or you could add an additional copy of UBE3A from using the viruses, for example, that I told you about with the mouse study. So we have those 2 approaches to turning back on UBE3A. Now the benefit to that approach is that that would be the upstream thing that's wrong, and everything that's downstream of UBE3A we hope would take care of itself. But the other thing we can do is we can try to look for things that UBE3A does or proteins that it modifies and go after those as our targets. And so, for example, we could try to find all the target proteins that UBE3A interacts with and see if any of those are proteins that already maybe have drugs available to modify their abilities and their activities. You'll hear some about that today. It turns out, we talk about how there's no UBE3A function in Angelman syndrome, but it turns out the researchers have seen that there's actually a very small amount of UBE3A that's expressed in the neurons in Angelman syndrome. The paternal copy may leak a little bit. So what if we could figure out a way to stabilize that little bit and keep it around longer, and we could put some more UBE3A into the neurons? And I just want to mention that one of the first postdoctoral fellows that we funded, and this was the Christina Castellana postdoctoral fellow, Jason Yee, just published a paper this year in Cell, which is one of our top journals in the science field, and he had found a modification of UBE3A that's important for its stability. So the money that you guys have raised and helped us with has already started some important work towards trying to figure out how to stabilize UBE3A. So here's another way that we can do this. All right, so a little bit of a preview for what you're going to hear today. And some of these people should be in more than one column, but I wanted to simplify it a little bit. We're going to talk about restoring UBE3A, and you're going to hear from the FIRE team, David Siegel and Kevin Nash specifically, but also Ed. Jody Cook is going to talk to us from Agilis Pharmaceuticals. Art Bodette and Ben Philpott aren't here, but I just want you to know that they fall in this group. So ISIS Pharmaceuticals is talking about the antisense technology to stop that antisense transcript and let UBE3A be expressed. And then Ben Philpott had published a paper with some chemicals that turn on UBE3A. Those chemicals are really harsh, but it gives us the proof of principle that there may be other drugs out there that could modify expression of UBE3A and be therapeutic for us. Thinking about modifying what's happening in Angelman syndrome and making those neurons function better, you're going to hear from a lot of people here. Jeremy Levin and Matthew During are coming up next from Ovid Therapeutics. You'll hear from Scott, Ann, and Ed from the FIRE team about some of the work they're doing that sort of falls in this realm. But I also want parents to know this, because we talk about our therapeutics possible. We already have therapeutics. And you're like, what? We have them already. We use them every day. We're talking about finding better therapeutics. What do I mean by that? We have anti-epileptic medicine. We have the ketogenic diet. We have the low glycemic index treatment. Those are therapeutic interventions for Angelman syndrome. They're targeting the epilepsy. And we're going to hear from Donna Herber. I think this is disruptive nutrition, not skivation, so I apologize. Today about a potential dietary approach. We know there are some behavioral therapies and medicine that can help with sleep issues. I'm happy to talk to anybody about this if you want to talk about the trial that was done that modifies behaviors in sleep. We can treat reflux with Prevacid. We can treat behaviors with therapies. We can treat motor issues as best we can with physical and occupational therapy. And then communication has really exploded recently with the advent of the more affordable iPad for those of us who can't prove that our children are smart, even though we know it's true. And I want to point out to you that tomorrow the educational symposium starts at 9 o'clock, and we have some of the leaders in those fields who are going to be here to talk about communication and some of the things that are possible with literacy for our kids. So I strongly recommend that you don't miss that. Okay, and with that, I just want to finish up by showing you how fast some of this has gone. So I told you that from 1965 until 1997, it took us that long to figure out that it was UBE3A. In 1998, so just 1 year after we figured out that this was the gene, the mouse model was created. Because once you know the gene, then you can go into the mouse and remove it and say, okay, now I have a mouse with Angelman syndrome. In 2007, this is the paper from Ed Weber's lab that showed that you could rescue that mouse model. This was the motivation for founding FAST. So this was why we came together and put this foundation together, because we said if that rescue happened, then there should be more money put into this because we need to be looking for things right now. 2011 is the paper published from Ed's lab showing that you could virally replace UBE3A. That same year, paternal UBE3A expression was turned on in neurons by Ben Philpott's lab with topotecan. 2015, Art Bodette's lab has published the paternal UBE3A expression activity by these antisense oligos. And now we're here to hear all the great science today. But just look how fast things have gone since the rescue of that mouse model. So it's been 8 years, and that might seem like a long time, but if you think about how long it took to figure out that this was the gene, we are now moving at a very rapid pace. And that's thanks to a lot of you sitting in this room who have done everything from bake sales to Ruby Tuesday dinners to things like that. The more money we have to put into the research, the faster this timeline is going to speed up. And so that's the end of what I wanted to do for the introduction. Thank you. And that's going to put us right on time. But we're happy to take a question or two while we mic up our next speakers if anyone has anything. So what we have, oh, I should mention this. What we have mics in the aisle. We'd ask you to come up to the mic if you have a question. And There was another thing I wanted to say. Oh, yes, I want to say there's no internet access in this room in general. We're sorry about that, but we have to keep the feed open so that we can do the live feed, and if we start to jam it up, it's going to be a problem. So we're sorry that there's no internet access, but we're hoping you're paying attention up here anyway, so that shouldn't be an issue. Sharon. Can you bring in program?
Oh, I was just curious. My daughter got our diagnosis early. She was born in '89 and had our diagnosis in like '92, and then the internet, happened, and it's not really all that important. But there was talk about when the mutation occurred. Either like right at conception or during or in utero, or was the egg mutation already in the egg? And I was just curious. Does anybody really know?
So our best guess with this, so is your daughter a deletion?
Deletion positive.
So deletions happen when the eggs are forming, and it happens during the process of copying chromosome 15 over and over again. So there happen to be regions on chromosome 15 that are absolutely identical to one another. So if you think about the chromosome as a zipper, what happens is it comes apart, you copy it, so now you have 2 copies, but those 2 copies have to sort of zip back together. What happens in Angelman syndrome is those identical regions mispair, and there's a little piece that's kind of looped out, and that just gets tossed. So the big point here is that it is something that happens in our cells pretty regularly. It is not something that anyone in this room did or looked at or ate or walked across. And in fact, it's possible that that deletion occurred while you were in utero.
Oh, okay.
So because you start to make the eggs, your own eggs, while you're still a baby developing. So some of that could have happened before you were even born. Okay. So there's no one in this room that should listen to anyone who tells them that they did something that caused that deletion. It doesn't happen. Similarly, point mutations that occur that affect UBE3A or change imprinting center defects, things like that, those things happen in our cells all the time. They just do. What's great for us in some ways, if you think about it, is that these things are not incompatible with life. We have our kids, and we love them. And we know that the life expectancy for Angelman syndrome is good if we can get them through some of the conditions they have, like the seizures, and we can protect them from danger. So, the cells do everything they can to survive and make sure that these individuals make it. Okay, so give me just a second, and we'll bring up our next team from Ovid Therapeutics.